Chapter 1, An Introduction to the Signs of Life. In this chapter, we will talk about the properties of life, levels of life, the process of signs, and how scientists try to control for variables in an experiment. We'll talk about scientific thinking, tables and graphs, and major themes in biology. The opening question for this chapter is, imagine that you're a future explorer, and during a space voyage, your team identifies a planet that has conditions favorable for life. Your first samples are microscopic, but you return some interesting results, including objects with regular shapes. So then the questions are, what determines if these objects are alive, and what properties will you use to say that they are living? Well, when we talk about life, we have to recognize it. So all life shares some shared properties. So the questions are, what are these shared properties? One of the first properties is reproduction. All organisms reproduce their own kind. And in this slide, you can see a mama elephant and her baby. Elephants produce little baby elephants, so they produce their own kind. Another property of life is growth and development. Information that is passed on from one generation is carried on by genes, and these genes control the pattern of growth in all organisms. All life requires energy. Every organism takes in energy and converts it to useful forms, and it also expels energy. So here in this slide, you see an elephant foraging. There's also order to life. Each living thing has a complex but well-ordered structure, and here we focus in on the elephant eyeball. All life is also made of cells, and some living organisms have just one cell, and some living organisms like ourselves and elephants have trillions of cells. The cell is a fundamental unit of life. All life also responds to the environment, and here in this slide you can see the baby elephant taking a bath in a lake or a stream. All organisms respond to changes in the environment, and I guess it's a very hot day for the baby elephant. Many of these responses help to keep an organism's internal environment within narrow limits. For example, isn't it true that our internal temperature is 98.6, and when it's too low or too hot, we know that something's wrong with us? Properties of living things, the last one is evolution. Individuals with traits that help those survive and reproduce and pass those genes and traits off to offspring are driving evolution of population. And in this slide you can see a current everyday elephant, and on the other side, the right-hand side, you can see a woolly mammoth. So these are related, even though the mammoth is extinct. So over time, there have been changes. So that's why when we see the elephant today, it doesn't look at all like the woolly mammoth, but you can still tell that they're related. Okay, so the question is a virus alive? Does it have those fundamental properties that all living organisms have? So it says, use the criteria for living things and decide whether a virus is classified as alive. So here are some fun facts. A virus cannot reproduce on its own, and it's not composed of a cell. So you know it's missing some of the fundamental properties, so maybe we can already conclude right now that maybe it isn't alive. Okay, so it says here viruses do show order and they can evolve. And it says all viruses do not have all the properties of life. So for the longest time, this book and many other books used to categorize viruses as non-living particles, but I was just talking to several of my colleagues and they said that there was a conference of scientists, and now most agree that we can say that viruses are in fact living organisms, even though they may be missing some of the fundamental properties of life. Now, when we study life, we study it at different levels. My background is in agriculture, so when I think about studying life, I think about fields of crops of cotton or alfalfa, or I'm looking at an individual plant or insects maybe attacking the plant or nutrients for the plants. So we study life at different levels. So the largest level is the biosphere. So if someone's talking about global warming and it's on the news, then they're talking about studying a life at the biosphere level. Then the next level is an ecosystem. An ecosystem consists of living and non-living components. So we are living in the Chihuahuan Desert, so this would be a desert ecosystem. Then the next level down is a community. So a community consists of all the interacting populations in an ecosystem. So for example, here we see zebras and wildebeest and some kind of deer at a watering hole in a savanna in Africa. But the truth is, here in the desert, we could have jackrabbits and coyotes and birds all interacting with each other, and they would compose a community. Then the next one is a population. A population is a group of interacting individuals of one species. So instead of looking at like wildebeest, zebras, and deer, actually we're just going to look at one of those species. Maybe we could just be looking at elephants, or here in the Chihuahuan Desert, maybe we could be just looking at jackrabbits. The next level down is the organism, and that would be the individual. Here in this slide we see an elephant, but we could replace it with any organism. Again, it could be a jackrabbit or it could be a coyote that's living in our desert. And then organ system. An organ system are all those systems within an organism that make it function and make things work together. Life can also be studied at the organ level. When I think about the organ or even tissue level, sometimes I think about health. I always think about physical health. When you go to the doctor, they're looking at your individual organs to see how they're doing, or they're looking at your tissues. So an organ consists of multiple tissues that cooperate to form a specific task. And in this case, we have a heart, and its task is to pump blood throughout the circulatory system. Then we have tissues. A tissue is an integrated group of similar cells, and they work together. So you can have epithelial tissue that is a covering, whether it's your outer skin or the outer part of your internal organ. Then we have cells, and in this slide you can see a picture of a cheek cell. So that's what your cheek cells look like, and this is the fundamental unit of life. And remember, it's one of the important characteristics of life, that all life is at least made up of one cell. And then within the cell we have organelles. So what is an organelle? An organelle is a component of the cell that performs a specific function, like we could talk about ribosomes or the endoplasmic reticulum. So all of those are organelles found within the cell. Then we have the molecular level. So we're looking at molecules. And in this diagram, we see a diagram of DNA. We see the double helix, and this double helix is responsible for carrying out information. So a molecule is a group of atoms bonded together. And then lastly, an atom is the fundamental unit of matter. And there you have the nucleus with protons and neutrons in the inside, and then orbiting around that nucleus, we have electrons. In this next section, we're going to talk about how do we study life. So the opening question is, what is the secret to the world's best cookies? Are all chocolate chip cookies the same? How do they differ? There are some outstanding chocolate chip cookies out there, but what makes a great cookie? How do chocolate chips vary, and how do some taste better than others? So scientists began finding out these answers by observing the world. So an important part of accumulating evidence is actually the scientific method. And the first step to the scientific method is observation. So scientists learn about the natural world by making variable observations, taking measurements and gathering data. Observations lead a scientist to ask questions about the world, like what makes a great chocolate chip cookie? Okay, so some chocolate chip cookies are better than others, and here in this slide you see a stack of very delicious chocolate chip cookies. The next step in the scientific method after observations is hypothesis. I always say that a hypothesis is an educated guess, but I qualify it. It must be testable and falsifiable. In other words, you must be able to build an experiment around your hypothesis, and when you do and collect the data, you must be able to prove it false. So a hypothesis is a proposed explanation to a question that can be investigated. Observation and experimentation are used to investigate hypotheses. So we need a hypothesis to help guide us to develop our experiment. And in this case, the question is that will lead us to our hypothesis is what recipe makes for the best chocolate chip cookies? Okay, so it says here, switching from butter to margarine will improve cookies. Okay, so we always think of the scientific method as a series of steps, and oftentimes we see them as linear steps, but in reality, they're not maybe all linear. Here in this diagram, you see that observation, question, and hypothesis are linear steps. One comes right after the other. But then once you do the experiment, and you receive your conclusions, you may want to return to the hypothesis, giving it like a circular flow in the diagram. What is the difference between a hypothesis and a theory? Remember that a hypothesis is a proposed explanation for an observation. In other words, it's an educated guess that is testable and falsifiable. And here in this example, they're giving the example of endosymbiotic hypothesis. That means that the organelles inside of a cell actually came from bacteria. On the other hand, a theory is a much broader explanation, and it's well substantiated and comprehensive. And the example here is the cell theory, and that's why we see a series of cells. But the truth is over time, independently, different scientists in different locations over time have done experiments, and no one has been able to reject the hypothesis. And for this reason, we have a theory like the cell theory, that all life is made of cells, and all cells come from pre-existing cells. Another opening question is how does the term theory differ in science from everyday use? So isn't it true in everyday use, we almost use it like an educated guess, but the truth is it's something much more. My best analogy that I can give you, it's the closest thing that we have to the Ten Commandments, right? The Ten Commandments are something that is unchangeable. So keep in mind, in everyday language, we think it's speculation, but when we talk about it in terms of science, a theory is well supported, it's testable, and it's objective. So when we do an experiment, we have to do a controlled experiment. What does a controlled experiment mean? It means that everything stays the same except for your treatment. Only one variable is changing so that we can know exactly what's happening. And if we see that there are differences, we can attribute it to our treatment and not to any other factor in the experiment. So controlled experiment a controlled experiment means that everything stays the same except for the independent variable or the treatment. So we have these terms, independent variable and dependent variable. So what do they mean? Independent variable refers to the treatment of your experiment, and this can be manipulated and it can change. In fact, it is the only thing that changes in your experiment. On the other hand, we have a dependent variable, and the dependent variable is the response or the output. It's the effect. It's something that you measure. So the independent variable is your treatment, and the dependent variable is something that you measure. Now, within your treatment, within your independent variable, you can have both positive and negative controls. Most of the time we have a negative control. In other words, we have like no treatment. So a negative control is a group for which no change is expected. So oftentimes in class when we do an experiment, water will be the negative control. It's the no treatment. But then if we're using fancy equipment or something that we're not sure about, we'll also include what we call a positive control. A positive control is a group for which change is expected. So we're going to be looking at macromolecules. So if we want to know what the test is for starch or for fat, we'll put in a positive control where we know that there's starch in the sample, or we know that there's fat in the sample. And then when we get our result, we can tie it back to what we put in our sample. And this is called a positive control. Oftentimes, scientists run blind experiments. In other words, they withhold the information from the participants and the experimenter about which one is the treatment. And the reason why they're doing that is because they're trying to eliminate any bias. I remember when I used to work out in the fields, ag companies would come to me and say, could you please test this new product we've developed? We think it increases crop yields. Well, you know, I wanted the product to work, but the truth is that I was introducing bias into the experiment. It's much better if we do not know what the treatments are so that we do not introduce any bias. There's also something called the placebo effect. The placebo effect is like the sugar pill. You know how sometimes you give a patient a sugar pill and you tell them that it's medicine, and then after a while they start feeling better, but it's something psychological than actually physical. So the placebo effect is a phenomenon where you feel better, you feel a change, even though there is no treatment. So we also try to avoid the placebo effect. Another opening question: what observations can you make about the natural world? So we don't have to travel to exotic places like Africa, even though I'd love to go there. We can look around at our own beautiful Chihuahuan desert. So do you guys have any observations about the desert? Okay, so scientific thinking can be distinguished from other ways of viewing the world. So what is the difference between a scientific view of the world and something that is not scientific? Science is one way of knowing the world. It has its own hallmarks and it has its own limitations. Pseudoscience is the field of study that it is falsely represented, and it has oftentimes a false idea of a scientific basis. So how can you recognize pseudoscience as opposed to real science? Well, pseudoscience doesn't follow the rules of the scientific method. Usually pseudoscience does not go through results and repeat the experiment to see if they get reproducible results. They have unprovable or untestable claims, whereas in science, claims are proven and disproven. In science, all results are open to outside review. In pseudoscience, there is no review. And in true science, there's multiple lines of evidence, but in pseudoscience, there's really just a small amount of data, and underlying causes are typically not investigated. So we need to make a difference or draw a line in the sand between features of true science and that of pseudoscience. The scientific process depends on peer review. So what is peer review? Peer review. Peer review is the evaluation of work by impartial, qualified, and often anonymous experts who are not involved in that particular experiment. In other words, when oftentimes scientists submit their articles to a journal, there's a panel that actually reviews their manuscript. Through peer review, outside authorities can help verify the validity of scientific results. So how can you recognize reliable sources? Oftentimes we look on the internet, are those reliable sources? And sometimes that's really tricky business because you don't want to go to a source, especially if a company's been paying them, right? Because they're there to promote a product. They're not there to promote fair and unbiased results. So how do you recognize reliable sources? So there are questions that you can ask yourself to help you judge whether a source is a good source or not such a reliable source. Okay, so here are some questions, and this will help you. Is the information current? Is the source primary? Is the author or authors identifiable and well qualified? Does the author lack potential conflict of interest? Are references cited? Are any experiments described in enough detail that they could be reproduced? Was the information peer-reviewed? Is the information unbiased? And is the intent of the source known to provide information? So what is the difference between a primary and secondary source? A primary source would be the scientists themselves or reading their manuscript in a peer-reviewed journal. What's a secondary source? A secondary source is a description or a review of primary sources, often containing commentary. So we often hear secondary sources on the news. They'll say, a new study just came out. A new study that will help you lose weight and look younger. I always listen to those. Those are secondary sources on the news. But if I actually go to the Annals of Internal Medicine and I read an article on antibiotic resistance, then I'm going to a primary source. So oftentimes when we're doing an experiment, we have to look at the data and we have to analyze it. And oftentimes that means putting data into graphs. So here in this slide, you see some cookies, and you also see a graph of cookie A and cookie B. So there's a cookie bake-off, and it looks like cookie A is winning. The number of times chosen as a winner is 80 times, whereas cookie B is a little bit less than 20 times. So definitely cookie A is a clear winner. So scientists communicate their data using tables and graphs. So when you give your group presentations at the end of the semester, you too will use a table and a graph to represent your data. A scientific table is an efficient way to present a lot of data in a small amount of space. Graphs are means of displaying data visually, which can help to summarize and compare information. In this slide here, you see a table with a lot of data, and you see some headings and some subheadings, and you see it on the top and on the side, and then you actually see a lot of data within the table itself. In this slide, you see a graph, and this one is a line graph. It has a blue line and a purple line, and you see that the blue line is for men, and the purple line represents women, and the x-axis has years 1930 to 2007, and the y-axis says deaths per 100,000 Americans. So you can see that the death rate is much higher for men than it is for women, most all years except for 1930. Here you see a bar graph to describe lung cancer between different types of drugs. So here you see them represented in blue and red graphs, and these are for people under 70 and over 70. And then this is a pie chart. This is another type of graph. And here you see a circle, and it's divided up into pieces of pie. And the largest one here represents 57%. And this one is trying to describe lung cancer. So it's describing lung cancer in different ways within a pie chart. Another opening question is how can we explain both the unity and the diversity of life? So now that we've talked about the fundamentals of life and how we observe life through the scientific method, I guess the next logical question is how can we explain both the unity and the diversity of life? So they're all things that life has in common, and then you should be saying to yourself, oh yes, it's those fundamental properties that we talked about at the very beginning of this chapter. But then the other question is how do living things differ from each other? So in this slide here, we go back to seeing an elephant, and it says, if we look at some of the major themes that underline the study of biology at all levels, we can take a look at evolution, structure and function, information flow, interconnections, energy, and matter pathways. So all of these are sort of streaming around this elephant. The theory of evolution through natural selection is biology's unifying theme. So we keep in mind is that when we talk about evolution, we're talking about change over time. What kind of change are we talking about? Change of traits. When so many traits start to accumulate and change over time, eventually we get a new species, but you don't necessarily to have a new species to have evolution occur. So here it says evolution is the descent with gradual modifications of ancestral species to modern day ones, and this explains the common characteristics throughout living things. So we can have change over time, and this is a gradual thing. So evolution through natural selection helps to explain the development of antibiotic-resistant bacteria. A dear friend of mine was just telling me that her mother's in the hospital because she has an infection after she had like surgery done on her knee. And so I'm hoping that she gets better, and I hope it's not an antibiotic resistant bacteria that is infecting her knee area where the surgery took place. Structure and function correlate at all levels of biological organization. In other words, form and function go hand in hand. So within a biological system, structure, the shape of something, and function, what it does, can provide insight to each other. So in this example here, we have what's inside our lungs, and these show all the arteries and little tiny sacs, and when we're respiring, that oxygen is actually absorbed and we have gas exchange. Living organisms regulate the transformation of energy and matter. All activities of the cell require energy and matter to proceed. The sun is the ultimate source of this energy. The sun provides the energy that drives nearly every ecosystem. Perhaps the one ecosystem that it doesn't drive are deep sea thermal vents. The information in all genes is encoded in an individual chemical language common to all organisms. Many inherited diseases result from improper information in the form of a gene. And in this slide here, we see Muhammad Ali and we see I think Michael Fox. Both of these gentlemen have Parkinson's disease, and Parkinson's disease is caused by a genetic disorder or mutation. There are many interconnections within and between the different levels of biological system. At each new level, novel properties emerge that are absent from the preceding one. The cell displays an emergent, which is a non-apparent in the individual parts that make up the cell. And by looking at that, we see a cell here on the right-hand side, and this cell as a whole is very different if we were just to examine the parts. So here's an appendix of image-long descriptions, and so it gives you, here are some descriptions that you can take a look at on your own. This is the end of chapter 1.